Hello, my name is Alex, and I'm on visualizing proteomics data for researchers along with Emma, Colette, and Eli. Today, we're going to present our dashboards that we worked on the last 10 weeks with the guidance of our stakeholder, Dr. Akshay Bareja, who's assistant professor of medicine at Duke. So here is the brief content of our presentation. Proteomics. So proteins are the fundamental executors of cellular function. They assemble framework of cells mediating cellular communication and catalyze biochemical reactions that convert nutrients into energy. Proteomics is quite similar to a more well-known study, genomics, whereas genomics is a study of the organism's set of DNAs, proteomics is a study of all the proteins. While DNA provides a blueprint for making proteins, it doesn't guarantee you that those proteins are actually produced and working. Proteomics fills this gap by showing which proteins are present and active, linking genetic instructions to real-world health outcomes. Proteomics allows spotting subtle changes that often signal disease before symptoms even appear. There are many real-world applications of proteomics. For example, in cancer research, if a protein has more phosphate tags than usual, it usually means that cells' growth signal are stuck in overdrive and could become cancerous. Spotting these overtag proteins helps scientists find enzymes causing the problem and design treatments that can turn those signals down. In the study of neurodegenerative disease such as Alzheimer's, proteomics reveals early indicators of neural stress by tracking abundance of synaptic proteins. If there is a measurable decline of those markers, the individual has higher vulnerability to these illnesses and gives critical window for intervention long before irreversible symptoms set in. In infectious disease research, proteomics work by comparing all the proteins in infected samples with those in healthy ones. This lets researchers spot changes in the immune system and match how severe is the illness or how well someone fights it off. Those protein shifts can act as warning signs of disease outcome and help vaccine developers to see which part of the immune response are most effective against pathogens. Proteomics experiments produce huge, complicated data sets that can be overwhelming if you don't know how to code. Here's unprocessed data sets that we are working with. It is a sample mass spectrometry data, and it is 70 kilobytes of pure text, and as you can imagine, it is difficult to even navigate. Without specialized application, creating clear informative visualization of protein levels and modification is quite challenging. Fortunately, our stakeholder, Dr. Akshay Bareja, provides us with basic applications that extracts necessary data from mass spectrometry dataset and performs necessary calculations such as normalization. It is up to us, however, to create an accessible application to let researchers with minimal coding experience quickly perform proteomics analysis and produce publication-ready graphs that clearly show statistically significant changes between groups. The application is designed to read dataset, apply filtering and normalization, and then carry out differential expression analysis. It runs a range of outputs, including sample distribution plots, intensity-density plots, PCA plots, heat maps, volcano plots, and different enrichment analysis. Dr. Akshay Bareja provided us with core analysis code, and we built a user-friendly dashboard around it to produce all the necessary visualizations. Throughout the process, we worked in close collaboration with our stakeholders to ensure the tool met the needs of researchers. And Eli will take it from here to explain some of the data analysis tools and visualization used in this application.
Yeah, so now I'm going to go over the measurements and plots fundamental to our app. So first we have relative occupancy, and that's the percentage of a protein that is modified at a site under specific conditions. And this helps researchers understand how cells work and respond to changes like disease or treatment. Next, we have protein abundance, and this is how much of a specific protein is present in the sample. And it tells researchers what the cell is doing. So for example, high levels of certain proteins may mean the cell is growing, dividing, or responding to stress. Next, we have relative abundance, and this measures the amount of modified peptides detected between samples. So it doesn't measure the exact number of modifications, just the relative proportions. And it's useful for similar reasons to protein abundance, but also gives a better picture for large-scale comparisons. And then normalization is the process of adjusting data to remove technical variation, such as differences in sample size or instrument parameters, allowing for accurate comparison and interpretation of underlying biological differences. Now, in terms of plots, we have 6 in our app, but for the sake of time, I'll go over the 4 most important ones. So the first would be intensity plots, and these are used to visualize densities of log2 centered abundance and occupancy values across all samples. So each curve represents one sample distribution of protein intensities, and then the x-axis represents log2 centered intensity values, and the log2 scale handles a wide range of values, while the centering aligns the data around 0. And then the y-axis is the density, so it shows how common each intensity value is within a sample. The purpose of this plot is to check whether normalization works. So we can see how much the curves overlap with one another before and after normalization. And then if we conclude normalization did work, we can confidently assume that any variability in the data is due to biology rather than systematic error. Next, we have PCA, or principal component analysis plots, and these are used to visualize patterns in these complex proteomic data sets. So each point on the plot is a sample, and basically the closer they are, the more similar they are. So PC1 and PC2 are the axes, and they represent the directions of greatest variance in the data, and they're identified through principal component analysis, which is a dimensionality reduction technique that extracts the most important patterns in a data set. And the purpose of this plot is to see if different genotypes show distinct protein expression patterns, and it also allows researchers to easily identify outliers. Next, we have heat maps, which are used to visualize protein or relative abundance levels across samples in a color-coded matrix. And they quickly show patterns like up or down regulation across conditions and help researchers identify clusters, trends, and differences in expression and modification profiles across the data set. Lastly, we have volcano plots, and these are used to highlight the most significant differences between two experimental conditions, typically control and treatment. And each point represents a protein, and the red points you see represent the specific type of protein that you have the ability to select from a dropdown in our dashboard. And the x-axis represents how much protein abundance, or relative occupancy, depending on the specific plot we're dealing with, changes between the two conditions and in which direction. So positive values suggest higher levels in the treatment group compared to the control, and negative values suggest the opposite. Then the y-axis indicates how statistically significant the observed change is. So higher values indicate stronger evidence that the change is real and not due to random variation. And this plot's really important because it allows researchers to quickly spot which proteins had measurements that both changed a lot and with a statistically reliable change. And now I'm going to pass it off to Emma to talk about the starting point of our app.
Thank you. So now I'm going to introduce the version of the dashboard that Dr. Bareja provided us with that we were tasked with extending and improving. So first, you have the home page. There's no description of the app, so it's unclear what functionality the app provides. And there are buttons that you can't utilize yet because you haven't uploaded the two data sets. Once you do upload the data sets, there's no start analysis button and there's no processing indication. So after 20 seconds of waiting, this table for relative occupancy appears. However, you can't scroll horizontally, so multiple columns of the table get cut off, and there are no tables for relative abundance or protein abundance. Here, the plots tab was completely empty. And for the differential expression analysis tab, again, only relative occupancy data is displayed, and the volcano plot is at the bottom, so unless you scroll all the way down, you don't know that it's there. I'm also going to talk about our process of building the application. So we used a Google Sheet to keep track of our different requirements, so we knew exactly what tasks we had remaining and their respective priorities so that we could allocate time effectively. We also shared this with Dr. Bareja so he could add any comments. Now I'm going to talk about our tech stack. So we developed our app as an R-Shiny application, and it's packaged using the Golem framework, which helps make Shiny apps modular and more maintainable. The source code is hosted on GitLab, and we've set up CI/CD pipelines to automate testing and deployment. The app is also deployed to Ubuntu Linux virtual machine using Docker containers for consistency and portability. Now I'm going to talk about the challenges we faced. So one big challenge that we faced was changing goals. So multiple situations happened when after we implemented a feature, we realized that it would be useful to modify the feature or have other features as well. For example, we initially implemented heat maps as only displaying the top 25 proteins or top 25 sites. But then after having a conversation with Dr. Bareja, we realized it'd be very useful to have the users to be able to choose to display the top N proteins or sites. Another challenge we faced was understanding the science, as initially we didn't really understand the science behind the plots, so we weren't able to explain their function or purpose effectively when asked about them. Another big challenge we faced was switching to Golem, as we needed to restructure the entire code to be modularized. Lastly, using Git meant that we inevitably encountered merge conflicts and ended up with some features going missing here and there. Next, I'm going to pass it on to Colette to give you a demo of our current app.
Thank you. This is what the final state of our application ended up as. So first we have our home tab. We have an explanation of the app for new users. Here I'm uploading the proteins and the peptides data sets. Both are necessary for the analysis, and only once those are uploaded does the submit files button appear. We also have processing indicators, so you know the app is working, and we have a file summary at the corner, viewable at all times. And once it is done, we have an analysis ready message. We also have an upload history section and a sample data set section in case a user wants to try the app but doesn't have any data to use. Next, we have our normalization tab. Now the tables are scrollable. We've also shortened the column names and decimals to 4 significant figures. We also have select columns option in case a user wants to focus on specific columns. We have this table for relative abundance, protein abundance, and relative occupancy. And so all of our other plots have all 3 as well. Next, we have our normalization plots. We have a before and after intensity and box plot for each. As you can see, we know normalization has worked because they're both more closely aligned and overlapping. The effects of normalization sort of cancel each other out for relative occupancy, so there's no change here. Next, we have our principal component analysis. They're very interactive in that you can hover over any point to view the exact sample name. The group names are dynamically generated from the data as well, and they're colored and shaped for accessibility. Here are our heat maps. We have a colored row at the top indicating group. We can also choose exactly how many proteins to show, up to 100 and down to 10. We can also customize how the heat maps look to whatever height and width we would like, and that changes how it's generated down here. And we can also click this option to show the full heat map without any borders. Next, we have our differential expression analysis. We have a volcano plot and a table of values for each, which is now scrollable. And we can choose what gene to highlight in red on the volcano plot here. And the highlighted genes data is also showed in the selected extension tab down here. We can also change the comparison direction if we would like. Next, we have our gene set enrichment analysis. Here we have to choose what species to use. And then we can select which pathway, which is a collection of genes with a shared purpose we would like to view. And additional information like the enrichment score, the p-value, and the pathway size can be found down here. Next, we have our kinase substrate analysis. We first have to upload a kinase substrate dataset, has the same logic as the first dataset upload, and then we have to select what species we're looking at. We can see the z-score for all of the gene exceptions in this graph. Additional information can be found in these 2 subtabs as well. We can't conduct this analysis on protein abundance since it is relies on peptide data. Some other features that we have a download and save buttons for the data tables and the graphics in our app. We also have help buttons on each tab that explain the purpose of the analysis being done on that tab. We also have show code buttons that allow the researchers to alter the calculations if they'd like and to ensure replicability. We also have a summary tab that gives the highlights of the analysis results found. And lastly, we have a restricted access admin tab that allows developers to monitor use of the app. Thank you. So that's the end of our presentation. These are 2 links to our website and our GitLab repo if you're interested. Does anyone have any questions?